Now, we talked about how overexpression of proto-oncogene can lead to cancer. Now let's take a look at tumor suppressor genes. For a tumor suppressor gene to lead to a cancer, that gene has to lose its function, right? So you have loss of or silencing of a tumor suppressor gene that is going to lead to cancer. And remember that the tumor suppressor genes can be compared to the brake pedal in a car, right? Or a truck. So whenever you hit the brake pedal, the truck stops or the car stops, right? Whenever the tumor suppressor gene is expressed, this is going to cause two possibilities. One is the cell is going to stop cell division. Two, it's that if there was already too many cells, this is going to trigger cell death, programmed cell death, or apoptosis, right? So, a mutation that causes the loss or silencing of tumor suppressor gene leads to leads to cancer. So some examples are going to be the Wilms tumor, right, where there is actually a mutation in the gene that is going to cause cells to overgrow, and specifically the cells in the kidney tubules, right, that are actually going to overgrow. Then the retinoblastoma. Retinoblastoma, it's a type of cancer of the eye, right? That cancer of the eye is caused by a mutation that occurs in the RB gene. That RB gene normally, right, in its normal function, it's to actually prevent the overproduction of some genes in the eye, right? So the retinoblastoma is observed in about one infant out of 20,000. So 2-point mutations are needed in the RB1 gene for cancer to be expressed, right? And these mutations can occur in germline cells or in somatic cells. Most of the time, though, when a mutation occurs in a germline cell, there is another mutation that is going to occur in a somatic cell, right? So a good example of this we are going to see in chromosome 13, where a mutation occurs that leads to cancer cell. All right. So retinoblastoma in an infant here, right? We can see actually the dark spot in the eye of the child that actually are the characteristic of retinoblastoma in a person. Now, what is amazing? Well, amazing. What is really incredible with today's technology, it's that retinoblastoma can be cured. Okay, it can be cured by doing some surgery. All right. So here I want to show you how the gene RB or its product normally works. So I was telling you about a transduction signal pathway. This is a transduction signal pathway. It's a cascade of events, right? I'm not going to bug you with the details. So let me spare you with the details. But what you need to know is that, typically a cell is going to receive a growth factor signal. That growth factor signal is going to lead to a cascade of events here. I was telling you about tyrosine kinase for the proto-oncogenes. So this is the kind of things you are going to see. So the phosphorylations. And then you have one RAS protein that activates another protein that is called RAF, right? And another protein that is called MEK, right? And another protein that is called ERK. Now, I want to show you the interesting thing. Let's go back here. Nope. Right. Nope. Not yet. Okay. So let me change the color here so you can have a good appreciation. Okay. So here we have RAS, KRAS, right? Here we have RAF, right? Where is another one? Okay, so another RAS right here, okay? Just to show you actually how, this can stimulate gene expression, but then they don't work alone, right? They don't work alone because the signal comes, right? But that signal is modulated. It is controlled. Not when the signal comes, the cell is going to systematically multiply, right? Well, what happens is that the cell actually has RB protein right here. That RB protein, let me change the color. That RB protein that is right here, its normal function is to attach to E2F. By attaching to E2F, that RB protein is preventing E2F from going and stimulating the expression of some genes, including the cyclins proteins that are involved in triggering the S phase. Now, the S phase is not triggered unless a cell is committed to cell division, right? So RB prevents the cell from moving on to the S phase, right? So now, when RB is inactivated, now E2F is going to be left alone. And when E2F is left alone, now E2F is going to cause expression of some genes that are going to lead to the production of proteins called cyclins that are very important to trigger cell division. So this is how a mutation in RB is going to lead to cancer, right? So, but for that to happen though, you need what we call a two-hit hypothesis, right? Which means that two mutations have to happen in the RB gene, okay? And these two mutations are going to cause inactivation of RB, okay? So another tumor suppressor gene is P53. P53 has two roles in the body. One is to cause the stop of cell division. The second role is to trigger apoptosis, right? So anytime this gene is going to lose its function, right, you are going to have cancer situation. And actually, many cancers are associated with P53, right? As much as 50% of human cancer involved an abnormal P53, right? If you go back to the previous slide, you are going to see exactly what I'm talking about, right? In a case of the Lee-Fromeni syndrome, right, we have P53 that is involved here, okay? P53 is a central gene that controls, that is involved in so many cancers, including the cancer of the bladder, blood, brain, breast, colon, esophagus, liver, lung, spleen, thyroid, you name it. There are many cancers associated with mutations that occur in P53. Now, question, what could trigger these mutations in P53? Well, this changes can be triggered by viruses, so environmental factors, including viruses, toxins like smoking, right? And radiations, to name only these three. These are not the only potential triggers, right? But these are very common triggers, right? That are going to happen, in somatic cells. So, you can also have some mutations that are going to happen in other genes. Then this is going to trigger some modification in P53 gene. All right. So here we have the example of this stomach cancer that occurred in one particular family. And in this particular type of cancer, ecadrins, which are proteins that are found in tissue lining, is going to play a very important role in the case of stomach cancer here. Now, you know that the stomach is has some tissue lining, as very specifically epithelium lining this organ. So in this particular type of cancer, the cancer was diagnosed in dad right here, and very quickly it was found that the cancer was a family issue, right? So that cancer actually was found in many members of the family, not just immediate family, but also related families, including cousins, right? Many of the family members actually, to prevent cancer development or to anticipate, well, they spend their life without actually a stomach. So knowing the history, well, they undertook the step to remove the stomach altogether so that they never really experience cancer. So this is the case of Bradfield family. So many members actually lived without a stomach. So they had to eat small bites at a time, but the trade was probably worth it when you know that a cancer would definitely lead to death, right? So in the case of breast cancer, we have some family cases of breast cancer, but we also have sporadic cases of breast cancer, right? When there is a family case of breast cancer, this is going to be associated with a germline mutation, right? That is going to be passed down. When it is a sporadic form of cancer, this is going to require a mutation in two genes, one or two alleles, one you receive from mom and one you receive from dad, right? Which is very unlikely. That is the reason why most of this type of cancers are more germline mutations that actually are maybe one mutation, but the other allele is fine, right? But that allele can be mutated at some point, right? So you have a driver mutation, and then another mutation that can happen, right? One single mutation that actually can trigger cancer at that point. Okay? Only about 5% of breast cancer account for familial breast cancer. However, in the major cases, you have one allele that is already mutated while the other allele is fine, right? But through the lifespan of the individual, if that normal allele gets mutated, this is when the individual develop breast cancer, and this is what happens most of the time. Okay, and most of the time, the mutations are going to involve DNA repair genes. Now we know today that BRCA have been associated with breast cancer, including BRCA1 and BRCA2, right? So this BRCA1 and BRCA2 are not only associated with breast cancer only, they have been associated with many other cancers, including the colon cancer, in the case of BRCA2, BRCA2, kidney cancer, prostate cancer, pancreatic cancer, gallbladder cancer, skin cancer, stomach cancer, and so on. So, having identified a particular gene and associated with a particular cancer, such as BRCA gene, doesn't mean that they cannot be associated with other cancers. That is very important to remember. So, the BRCA mutation are occurring in autosomes, right? Okay, they are occurring in autosome, and because they are occurring most of the time in autosome as dominant mutations, these are occurring with what we call incomplete penetrance. And as a reminder, incomplete penetrance means that two individuals with a mutation in the gene are not going to have the same phenotype. That's what it means. Some are going to have a severe disease, others are not going to have a severe disease, they're going to have a mild disease or not express at all, right? So we have a spectrum in a population that varies, right? So there is a difference in incidence in different population. That's what you need to understand. So BRCA1, you can see that BRCA1 is central, and that BRCA1 is associated to so many other things, right? Including BRCA2, RAD51, P53, RB1, CHEK1, HDAC, HDACS, right? So they can be associated with DNA repair, checkpoint for DNA repair, tumor suppressor gene, checkpoint regulation for mitosis, chromatin remodeling, misfolding of protein, okay, response to misfolded protein, and so on. So, environmental factors can involve, viruses, can involve toxins, can involve radiations, right? So all these conditions can actually trigger loss of apoptosis, loss of DNA repair, loss of control of the cycle, in a sense that too many cell divisions. Okay, so now, how do we diagnose cancer and how do we treat cancer? Well, the diagnosis of cancer has tremendously changed. it has gone from doing a whole biopsy to actually just isolating some blood and looking into blood cells and going to look into the genes, right? So it is tremendous how the approach for diagnosis has evolved. But a lot of the diagnosis still being done these days is going to use antibodies, right? Antibodies that are looking specifically at cancer antigens, okay? So the diagnosis and treatment of cancer have evolved tremendously, right? We have new approaches that go beyond identifying simply oncogenes and tumor suppressor genes, right? They are doing sequencing of genomes, right? Sequencing of exomes that allow to have a more targeted knowledge, a more targeted treatment. Now here we have a way of actually diagnosing cancer, and this way of diagnosing cancer allows us to see how cancer cells are different one from another and how we can actually look at their gene expression profiling, okay? So we have ALL, MLL, and AML. And so these are different cases of cancers. And these different cases of cancer, we are looking at the profile of gene expression in these cancers. And we are comparing them, right? We are comparing them and notice that, well, there is actually a huge spectrum in the variation. We go from the red here to blue here, right? From red here to blue here, okay? And so you have a standard deviation in gene expression that varies tremendously. And because of that tremendous variation, we are going to be more targeted in terms of how to approach the treatment as well, right? So the oldest treatment that is still the best treatment, it's going to be surgery, okay? So surgery is still by far the most effective treatment, right? Nevertheless, that surgery is not always possible because of the location of the tumor, right? What if we are dealing with a lymphoma? If we are dealing with a lymphoma, how do you deal with that, right? A leukemia. A leukemia, it's cancer of the blood by definition, right? How do you target specifically cancer of the blood? That is difficult, right? So then we are going to have other treatments, including chemotherapy, right? And radiation therapy. So these two are going to target, hopefully, cancer cells. Nevertheless, these treatments are not as specific as surgery can be, right? So the newer approaches are going to try to specifically target the mutated genes. So that, by going back here, by looking at the pattern of gene expression in a cancer cell, we can actually say, okay, well, let's specifically target this particular gene. The difficulty, though, it's that cancer cells are cells in our body that have 99.999% related to everything we have in our cells. Therefore, it becomes difficult to treat these cells when it comes to therapy, even that radiation therapy. But with that being said, radiation therapy is going to specifically target the mass of cells that are affected, right? That's the ideal situation. Nevertheless, practically, we see that, well, the cells that are neighboring cells to the targeted cell can also be affected, and the very principle by which we are destroying these cancer cells with radiation therapy, this very same principle is affecting our normal cell and can sometimes actually make the situation worse than it was at the first place. But, these are things that are good to know and for your information. So in treating cancers, we can interfere with the rate of cell division, right? By targeting, for instance, kinases, right? And cyclins, which are both involved in accelerating cell division. Some examples of targeting the rate of cell division is the use of mTOR inhibitors. Okay, these mTOR inhibitors are going to interfere with the activity of the serine threonine protein kinases, which are proteins that are involved in the signal transduction pathway that triggers cell division. CDK4/6 inhibitors are going to be these inhibitors that are targeting kinases and cyclin-dependent kinases, right? These cyclin-dependent kinases are also involved in the signal transduction pathway, which again is involved in cell division, okay? So CDK4/6 inhibitors have been successfully used in treating breast cancer, for instance. So checkpoint inhibitors are other ways to treat cancers. So the checkpoint inhibitors can be actually be used to 1, 2, 3. The immune checkpoint inhibitors can be, for instance, in the case of a monoclonal antibody that is actually preventing, right, the cell from undergoing cancer. How do they do that? By actually binding some receptor that we call PD-1, okay? And that receptor PD-1 is actually the antibody is coming and binding on PD-1, right? By binding on PD-1, this is preventing the signal to bind on that receptor, right? And by preventing that signal to binding on that receptor, there is no signal transduction pathway that is put in place, right? Therefore, cancer is going to stop that way. So more specifically, so we have, actually, I got a little bit confused earlier. Specifically, a cancer cell is going to have on its surface a PD-L1 receptor. That PD-L1 receptor recognizes PD-1, that is a signal molecule, and that signal molecule is attached to a T cell. Now, normally, PD-1 attached to PD-L1, right? But with the antibody coming, the antibody comes and bind on here. By binding on the PD-L1, now the PD-1 is not going to be able to attach to this cancer cell, okay? And by not being able to attach to a cancer cell, now this cancer cell is not going to undergo, multiplication anymore. So let me make sure that everything is fine here. All right, so we're good. Then we have the chimeric antigen therapy. In the chimeric antigen therapy here, we are going to design a cell that is modified, right? So we're going to isolate a T cell from a patient. That T cell is going to receive a modification. That modification is called CAR, okay? So that now modified white blood cell that is expressing a CAR stands for chimeric antigen receptor. That chimeric antigen receptor is going to be expressed in a genetically engineered cell. Now, after verifying that the T cell is expressing a chimeric antigen receptor, these cells are going to be grown in a lab. And after successfully growing the cells in large amount, now the cells are going to be put in a patient by infusion, right? So as the cells go in, right, the cells with the receptors are now going to recognize cancer cells, and by targeting cancer cells, the cells are going to die. So this is an example of chimeric antigen therapy. The good thing about that chimeric antigen therapy is that it's very targeted, right? Okay, and then we have, in terms of diagnosis, what we call a liquid biopsy. In a liquid biopsy, here, rather than obtaining a solid biopsy, we are just going to take some blood, right? And that blood is going to be analyzed for the presence of abnormal DNA. So we're going to detect cell-free DNA. This cell-free DNA has to be tumor DNA, because know that in our blood, we have a lot of cell-free DNA. And this cell-free DNA, it's basically our cells that die, okay? That die, why? For one reason or another, they do die, okay? Now, we're not just going to be interested in any cell-free DNA. We're going to be interested in cell-free tumor DNA, okay? ctDNA. And that ctDNA is what we are going to analyze and see that, okay, well, you know what? There is actually lung cancer genes, colorectal cancer genes, bladder cancer genes, right? We can detect that, okay? So not only the blood can be used, but also other aspects can be used, including urines, right? That can be used, sputum that can be used, feces can be used, and obviously, the blood. Okay, so in terms of liquid biopsy, what are the advantages? Well, one of the major advantages is that we can actually detect tumors that are just starting at the point where it is still asymptomatic, right? We can detect these tumors, okay? We can be more specific as to, what we want to target. Okay, we can monitor what is going on with the cancer and the treatment. Okay, we can monitor so many different things so that we are going to put the patient in a better spot, right? One disadvantage, though, is that we can have false positive because remember that, the DNA we are looking at, it's cell-free DNA, right? That cell-free DNA, a lot of it is coming from our normal DNA. So it's very difficult to tell apart, right? So that we can have a number of false positive. Okay? Um, all right. So here I'm showing you, for instance, here how you can isolate this circulating DNA, right? That DNA is circulating in the blood, so you can, get DNA, tumor DNA from, for instance, lung cancer, pancreas, colorectal cancer. You can identify all of that. So this is basically, what we are going to cover here, and which concludes our chapter and this course. Thank you for your attention. Bye now.